Alright, so far we've talked about monohybrid crosses and how to make gametes. Now let's consider the dihybrid cross. In a dihybrid cross, we're talking about a cross between two traits at a time. So for example, eye color and hair color. And we want to see, given two different adults, what the end result is of a cross between individuals with eye color and hair color. So how do we set up a problem like this? First thing to do is to go through and pick out the important information in the problem. Let's consider this example 2. It says that in fruit flies, normal wings are dominant over held out wings, and red eyes are dominant over sepia eyes. In this cross, we're going to cross a red-eyed, normal-winged fly with a sepia-eye, held-out wing fly. We also know that both flies are homozygous for both traits. Okay. Whenever we go out to set about solving a problem like this, let's go through our basic steps. Step 1, establish our key. Here we have the phenotypes, the physical expressions of the traits I'm interested in. Red eyes and sepia eyes. I'm talking about eye color, so I'm going to use the letter E. From our problem, we know that red eyes are dominant, so we're going to denote that with the capital letter E. Sepia eyes are recessive, lowercase e. In terms of wing color, I could use the letter W, but uppercase W's look a lot like lowercase w's, and I don't want any confusion. So I'm going to use N, capital N, for the normal dominant trait, and lowercase n for the held out wings, which are recessive. Step 1, check. Step 2, write out the parent phenotypes. In this cross, I know that I have a red-eyed normal-winged fly. Red, normal. I also know that this fly is homozygous for both traits. I also have a sepia-eyed, held-out wing fly who is also homozygous for both traits. I've now written out the parent phenotypes. Next, I'm going to go ahead and write out the parent genotypes, the actual alleles that they have. They're red and homozygous for red eye color. Homozygous meaning two of the same alleles. The allele for red eyes is a capital E, so they have two uppercase E's. They're also homozygous for the normal wings. Check my allele key. Normal wings are denoted by a capital N. There's parent 1. Parent 2 is sepia-eyed. Sepia, if I look back at my allele key, it's recessive. Remember that the only way to show the recessive phenotype is to have a homozygous recessive alleles. So lowercase e, lowercase e. Same thing for the wings, lowercase n, lowercase n. Here are my two flies. The next step is to write out the possible gametes. What are the different combinations of gametes I can have from this parent? Alright, this is where we need to stop and pause and really think about what we're doing here. How many traits are we considering in this cross? 2. We have the letter E trait and we have the letter N trait. 2 traits. So how many traits do I want in each of my eggs or each of my sperm? That's right. I want both of those traits. I want 2 traits in each egg and sperm. How many copies of each trait do I want? 1. I want to make a haploid gamete, which means there's only 1 copy of each trait in the gametes. So I want 1 copy of E, I want 1 copy of N. Let's consider what my possible combinations are from this parent. How many choices for letter E do I have? 1. I can have a capital E or a capital E. How many choices do I have for N's? 1. 1 times 1 is 1. So there's only 1 possible gamete here. And it is capital E, capital N. There's my egg. I can't split it up now. Let's consider this parent over here. How many choices of E do I have? 1. How many choices of N do I have? 1. 1 times 1 equals 1. My only possible gamete here is little e, little n. There's my sperm. I can't split it up. The problem wants to know what the offspring look like. So let's draw out a Punnett square and look at what my possible offspring are. In this Punnett square, I'm going to put the gametes, the different gametes from one parent across the top and one parent across the side. Because I only have one possible gamete from each of these two parents, I only need a 1 by 1 box. Okay, one possible gamete across the top, the other parent's gametes on the side. Now if I cross these and put them together, convention says the capital letter is always listed first. I have capital E, lowercase e, capital N, lowercase n. That's the genotype of the F1 offspring. The parents, a cross between two parents, gives rise to F1 generation. A cross between two members of an F1 generation gives rise to the F2 generation. Can you predict what an F2 generation would give rise to? That's correct, an F3 generation. Now let's analyze this cross and figure out what these offspring look like. They have one copy of the dominant allele for eye color. In order to show the dominant phenotype, I only need one copy of that allele. So these flies have red eyes. And they have one copy of the dominant wing type, so they have normal wings. Take a look at the alleles. They have two different alleles for each trait, making them heterozygous for each trait. What's the genotype that I've determined? The genotype is capital E, lowercase e, capital N, lowercase n. Now in a second we're going to take the problem a step further. These are the F1s. I want to see what happens when I cross the F1s with themselves. Alright, let's see what happens when I do this cross. I want to cross the F1s with themselves now. From the first part of the problem, I know that my F1s have red eyes and normal wings. And I also know they're heterozygous for both traits. The genotypes of my F1s are capital E, lowercase e, capital N, lowercase n, and since I'm crossing them with themselves, it's the same thing on the other side. Now, the question is, how many different gametes do I have? Let's analyze this diploid parent for just 1 minute here. Here's my parental genotype. Let's first of all figure out how many different potential gametes I can come up with. How many choices for E do I have? I have two different choices, a capital E or a lowercase e. How many choices for N do I have? That's right, two more. I have a capital N and a lowercase n. So I have a total of 4 different gametes that can result from this cross. Let's use the FOIL method. FOIL works really well for dihybrids. In math, you do the FOIL method, multiplying the first terms, the outside terms, the inside terms, and the last terms. Let's do this to figure out the gametes for this diploid parent. First, I'm going to do the first terms for each trait. So the first E, the first N. There's one possible gamete. Now I'm going to do the outside terms, E, capital E, and lowercase n. Now the inside terms, little e, big N, And now the last terms in each allele pair, little e, little n. Count them up. 1, 2, 3, 4. This checks with what I did up here. There are my 4 gametes. Remember, each parent had 2 copies of each trait. In the gametes, I want to make sure that I have representation of each of the traits, but I only want 1 copy. So now, here are my eggs. I can't split these up. These letters are stuck to each other. Since both parents are the same, these are also the possible combinations for the sperm. So let's go ahead and fill these in the designated area. If you go through these steps each time you have a cross, you'll keep yourself straight and you'll be able to have the correct information for your cross. Now I need to draw my box. Remember that I need to put all of the possible gametes from one parent down the side, all the possible gametes from the other parent across the top. Let's figure out how many rows and how many columns my box will need. If I'm going to put this parent down the side, I have 1, 2, 3, 4 different gametes. So I'm going to need 4 rows. And since I have 1, 2, 3, 4 different potential gametes from the other parent, I'm also going to need 4 columns. Let's go ahead and now fill in one parent down the side, all the possible gametes. Remember, these are now eggs. These are now sperm. I can't split them up. Each one gets its own row or its own column. The other parent goes across the top, each different genetic gamete in its own column. My next step is then to go in and fill in my boxes, do my crosses. Fill this in along with me and check your work. Remember, the dominant allele is always written first. All right, now that I've written out all of my genotypes, it's time to analyze my cross and figure out what everything looks like. The most useful way to do this is to start in the top box on the left-hand corner and determine what this individual looks like. Capital E's and capital N. Therefore, this individual is red eye, because there's at least one copy of the dominant red trait, and normal wings. So I'm going to go ahead and write out that phenotype over here. Red eye, normal. And I want to figure out how many look like that. So I'm going to assign red eye normals a little yellow dot. So this one looks red eyes and normal. Again, here and here, at least one copy of the dominant trait of each of those traits. Red normal, red normal, red normal, red normal. And if I count all of these up, I have 9 red normal offspring. Let's consider the next open box right here. This one has red eyes and held out wings because they have the double recessive for that. So let's figure out how many red held out flies I have. Let's assign them green. You can also do this with symbols. Red held out, red held out, red held out. So I have 3 flies that show that red held out, phenotype and genotype. Let's consider the next one. This here has sepia colored eyes because they have the double recessive and normal wings. So I want to figure out how many sepia normal winged flies I have. I'm going to give each of these just a little red dot. Okay, sepia held out, sepia normal, and sepia normal. So I have 3 offspring that have that sepia normal phenotype. Last but not least, my final box here have sepia wings, or sepia eyes, and held out wings. And there's just 1. So my phenotype ratio is 9 to 3 to 3 to 1. This is always the ratio I see when I'm crossing 2 heterozygous parents. 2 parents who are heterozygous for both traits under consideration. I always result in a 9 to 3 to 3 to 1 cross.